Elephants, mice, trees, and poppies all start as a single fertilized egg with the potential to divide and grow into the complete body of a new individual. To make the vast number of cells required to build individuals, new cell contents must be created, and then one cell must divide into two. This well-organized pattern of events is called the cell cycle. The cell cycle is split into two parts, interphase and division. We're first going to have a look at interphase. Interphase can be divided into three discrete stages: G1, gap stage 1, S, synthesis stage, and G2, gap stage 2. During gap 1, the cell functions normally and protein and organelle synthesis occurs such that the cell doubles in size. The extra proteins and organelles mean that both daughter cells will be able to function the same as their parent cell. In the synthesis stage, DNA is replicated. This is done by semi-conservative replication and produces two identical copies of each chromosome. Chromosomes are wrapped around proteins called histones in eukaryotes, and together with these proteins, chromosomes are called chromatin. After DNA replication has occurred, two identical chromosomes called sister chromatids are formed and held together at a point called the centromere. When DNA replication happens, it is checked for any incorrect copying which may result in mutations that stop the new cell working. Finally, during gap 2, more growth occurs and proteins are rapidly synthesized in preparation for mitosis. It must be noted that, during interphase, the individual chromosomes are unraveled. This allows access to the genetic material, enabling new proteins to be synthesized. By the end of interphase, the cell contains enough cytoplasm, organelles, and DNA to form new cells. The next step is to share out both the DNA and the contents of the cytoplasm so that each new cell can function independently. The DNA is separated in nuclear division, mitosis, and cytoplasmic division also follows this. Mitosis consists of 5 different stages. Let's have a look at them. During prophase, the chromosomes condense, becoming shorter and thicker, with each chromosome visible as 2 strands called chromatids. Apart from the occasional mutation, the 2 strands are identical copies of each other, which are produced by replication. They are effectively 2 chromosomes joined together at one region called the centromere. During prophase, microtubules from the cytoplasm form a 3-dimensional structure called the spindle. The centrioles move around the nuclear envelope and position themselves at opposite sides of the cell. These form the 2 poles of the spindle and are involved in the organization of the spindle fibers. The spindle fibers form between the poles. The widest part of the spindle is called the equator. The breakdown of the nuclear envelope signals the end of prophase and the start of the next stage. The next phase is called metaphase. The chromosomes' centromeres attach to the spindle fibers at the equator. When this has been completed, the cell has reached the end of metaphase. In the next stage of mitosis, anaphase, the centromeres split. The spindle fibers shorten, pulling the 2 halves of each centromere in opposite directions. One chromatid of each chromosome is pulled to each of the poles. Anaphase ends when the separated chromatids reach the poles and the spindle breaks down. This last stage of mitotic division is called telophase. This is effectively the reverse of prophase. The chromosomes unravel and the nuclear envelope reforms so that the 2 sets of genetic information become enclosed in separate nuclei. After nuclear division, the final reorganization into 2 new cells occurs. This is called cytoplasmic division. In animal cells, the cell surface membrane constricts around the center of the cell. A ring of protein filaments bound to the inside surface of the cell surface membrane is thought to contract until the cell is divided into 2 new cells. It has been proposed that the proteins actin and myosin are responsible for muscle contraction, and they may also be the proteins responsible for cytoplasmic division. Instead of undergoing this contraction, plant cells synthesize a new cell plate between the 2 new cells. Now we're going to look at the purpose of mitosis. Mitosis ensures genetic consistency with daughter cells genetically identical to each other and the parent cell. Each daughter cell contains exactly the same number and type of chromosomes as their parent cell. This is achieved by DNA replication prior to nuclear division, the arrangement of chromosomes on the spindle, and their separation of chromatids to the poles. This genetic consistency is important in growth and repair, and also in asexual reproduction. Mitosis occurs in the growth of any organism as it develops from a single cell into a multicellular organism. It ensures that a multicellular organism has genetic consistency. All the cells in the body have the same genetic information. Some organisms can regenerate lost or damaged parts of their bodies using mitosis. Starfish can even regrow a completely new body from a fragment. Mitosis also allows old and damaged cells to be replaced with identical new copies. So just to recap, we've learnt that the cell cycle consists of 2 stages, interphase and cell division. Interphase consists of 3 stages, G1, S and G2, which are responsible for protein and organelle synthesis and also DNA replication. Then we learnt that the cell cycle progresses to mitosis, which consists of 5 sections: prophase, metaphase, anaphase, telophase, and cytoplasmic division. These phases are responsible for helping the cell to divide into 2 new cells that are identical to each other and also its parent cell. Thank you for watching, and I hope you've learnt something about the cell cycle. Bye bye.